our next presentation is from the Exeter hub, but I don't see Krasimira.
I think I'm here. Can you hear me?
Ah, perfect. She's online. Okay, that's why.
Sorry.
Oh, thanks a lot, Krasimira. Can you hear me?
Yes, I can hear very well.
Okay, excellent. So, Krasimira, we very much look forward to your presentation of the Exeter hub.
Thank you very much, Carola. Thank you. Thank you for inviting me. And I'm kind of really sorry that I couldn't join in person. And I hope, I hope I'll be able to still update you on what we've been up to. It's a shame I'm missing the interactions, but I'm hoping again that we are going to be able to meet in the future, and we'll have follow-up events where we can discuss in person. So thanks again. I similarly to the presentations from the other hubs, I would like to give some updates, and I'm hoping that these presentations and these conversations will continue in the future and initiate further collaborations. Some have already started. Just very briefly to introduce the hub at Exeter. Again, there are already some common topics, but each of our hubs, as it becomes evident, has its own kind of niche and space. So in our hub, we're focusing particularly on uncertainty quantification, which I know some other hubs are also doing, and how it can help healthcare and biomedical applications. And I'll show some examples of the work that we've done in that area. And the second theme that we have is on well, it's written there, mathematical approaches to evolving microbial communities, but it's kind of more broadly focused in terms of application on infectious medicine and antimicrobial resistance. And again, I'm going to show some examples. What I'd like to point out that within our hub, the expertise in terms of mathematics and the interests are particularly focusing on dynamical systems modeling. So, and again, that probably will come through with the examples that I'm going to show. In this presentation, I have focused on, similarly to the presentations that we saw, on some ongoing projects and progress that we have made. Just before I move on to this, I'd like to say that we are a growing community of researchers and partners. And I don't have exact statistics right now, but we have approximately 20-30 researchers working in the hub or in close cooperation with the hub, as well as a number of industrial and clinical partners. These are some logos of some of our partners. And the work that I'm going to show you today involves some of these. So each of the projects involves either clinical or industrial partner. So, and the acknowledgements, I'm showing the pictures of my colleagues that have provided the slides and that have worked on these projects. So the first example is modeling Duchenne muscular dystrophy. It's hard to pronounce. Here we're focusing on patients' trajectories, and this is collaboration with industrial partner. It's a data science, health data science company at Exeter, Certus Technologies, and through them we have established collaboration with one of the hospitals at UCL. They have been working for a long time with a team in UCL. In the UCL hospital that is looking at this disease. So Duchenne muscular dystrophy, or DMD, is a genetic disorder. It's characterized by the progressive loss of muscle, and it is a very complex condition. It has multisystemic failure. It's characterized by multiple systems in the body failing as children develop. It is genetic, so children are born with this genetic condition, and they progressively, as they grow, their condition worsens. And it's really of interest for families and for patients to be able to, and also for treatments, to be able to have some idea of how the condition of an individual child will change over time. So this is the motivation behind this project. And yeah, so some of it's written here. So we wanted to develop a model that can predict how an individual patient is likely to progress. And like, again, with many health conditions, there is a huge heterogeneity in terms of how patients' trajectories evolve over time. The variability is really high, as it's common in real health data. There are missing values. These are measurements that are routinely done to patients, but sometimes they miss an appointment, or for some other reason they go sometime later. So the data is quite messy. The time structure is clear because it's based on appointments. However, sometimes there are missing data, and we want to include treatment, and we wanted the uncertainty to be accounted for. So, and that was kind of also touched upon how interpretable or explainable our models are. So we are aiming here to be as explainable as possible, so that it can allow clinicians and to kind of actually use it for in terms of decision support. So I have several slides that outline the idea here. It's a dynamic, it's a state, it's a model that evolves in time. So we basically are looking at predicting, in some sense, the observations or the patient measurements, given different clinical measures that are used to track patients' conditions. And we take into account several different parameters here, or functions that define this observation. So we assume that there is some underlying healthy state. There is a disease state as well on top of it. There is some individuality. We call it individual nuance. That's what the postdoc Evan has told it. And we are incorporating measurement error as well. The healthy state, as I mentioned, represents what the patient would be if they didn't have the disease. And this is information that is very hard to actually find or have. We have the disease state, which accounts for the various effects due to the genetic mutation. Then we have, and this disease state, I have to point out, potentially could be related to some physiological changes that occur in the body, although we are very far from that, from actually doing that, but it is a possibility. Individual nuance is basically allows for patient-specific functionality of this model, and then we have the measurement error. In terms of the parameters that control, so this is a statistical model. They are basically probabilistic. We have distributions that define patient states, but we also can tailor each prediction to a specific individual. So in that way, we share some information between patients, which is common, but also we have the personalized submodel. The healthy state now, just the various components of the model. In the healthy state, we have like that kind of linear function. The healthy state at time t depends on a parameter alpha. The healthy state at times previous time plus delta, which is related to an external factors affecting the healthy state. Alpha controls how this might be leveled off as patient matures, and alpha is basically placed a distribution, allows us to formulate a distribution over the values for the healthy state, and we choose here H20 in the specific examples that I'm showing. So with this model, we are tracking observations. This is an NSS-AA score, which is a clinical score that is a composite measure of different patients' characteristics at specific ages. But doctors are also interested in terms of these other types of patients' measurements, such as walk time, rise from the floor, and others. So very quickly, we have a function that models the disease. We incorporate natural decline and also the effect of treatments, which is of interest as well to clinicians, because these patients are treated with steroids and they have a lot of side effects that contribute to worsening of patients' conditions or children's conditions. So the nuance is a basically random walk that allows us to be flexible in the model. And here are some results. We basically, what we have done so far with this model, we are, so here in this graphs, we are predicting this clinical measure or score, which defines the patient state over time. And we are, the model is learning from a subset of measurements for a specific individual, for a specific patient, and predicting their outcomes in terms of that score in the future. So the blue points are kind of training points or points that we've provided to the model, and we are predicting the red ones. And we can see here 2 examples of 2 different patients, kind of illustrating again how different they are, and the model behavior along with some uncertainty measures here, bounds for these predictions. The green line is actually here corresponding to time at which these children in these specific examples have started taking steroids. The treatment effect is something that we're still working on. So this, like any other models, can be updated with new information and the predictions. If you supply the model with more information, the predictions become more accurate, or the uncertainty bounds shrink, the uncertainty decreases. And we are also can look at other types of clinical outcomes, such as walk time or rise from the floor. As I mentioned, these deteriorate over time. So what are we trying to do next with this model? Going forward, we're looking at number of improvements, potentially getting some information from healthy children, although this is, as I mentioned, is very challenging because healthy children are not followed in the same ways as children with DMD. Talking to clinicians is really important, and trying to incorporate as much as possible domain expert knowledge in this modeling, looking at improving the speed of calculations. At the moment, it's using MCMC, so it's a bit slow. And we want to apply these different types of problems, such as synthetic data, which we have started working on, and predicting treatment outcomes, and potentially applied to other degenerative diseases. The framework is very general, and it's about to be submitted for publication. So we're hoping that it could be useful in the context of other diseases as well. So moving on to another example, something that we've been working with one of our collaborators and co-investigator from Imperial College, Wojciech Dilo, who is actually formal co-investigator on the grant. Here we're moving to applications to endocrinology, and specifically looking at hormone dynamics, which is highly pulsatile. These are measurements of LH, which is a reproductive hormone. These are used in clinic to kind of diagnose and assign treatments for patients with various reproductive disorders, including IVF treatments as well. This information is used in IVF for developing IVF protocols, especially when the situation is more complicated. But what we have done in that space is developing I can call it semi-mechanistic model of the driver of this pulsatility, which is in the brain, and we are combining this with Bayesian inferencing techniques in order to basically predict clinical data, as well as derive some underlying information about the underlying mechanism in terms of what might be defining when there will be a pulse or no pulse in this hormone. As I mentioned, pulsatility is critically important for healthy reproductive function, and therefore there is an interest. This we have developed a web tool that is available online for clinicians to use as well on that project. Another work that we have started developing is in collaboration with the Exeter Center for Excellence in Diabetes Research, and it is taking advantage of the availability, and this is kind of, again, a rare data set of actual human islets. These are the groups of cells in the pancreas that secrete insulin, and the beta cells specifically in the islet secrete insulin. And disruption of this, as probably all of you know, leads to type 1 or type 2 diabetes depending on the various factors that are involved in the development of this disease. In this particular case, we have access to islets from patients with type 1 diabetes that have sadly passed away and have donated their pancreas. So there are samples available from these patients where our collaborator, Professor Noah Morgan, and his group are looking at quantifying how many and what type of cells are found there. And they've been doing this for a number of years. What the modeling can help here is try to reverse time and make some predictions about how the patient got to that stage where their conditions was already fatal. So to do that involves a lot of image analysis. Basically, our research fellow here, Nicholas, has joined us around about 4 months ago. He has started analyzing this bank of images and defining the morphology of these islets, extracting the identity of the different cells, and creating networks. It is known that these cells are connected within the islet. The idea is that we can use these individual patient-derived networks to superimpose on the mechanistic models, which we have been developing for a long time, to study the progression of type 1 diabetes. And here are some examples of benchmarking this analysis to something that has been previously published based on manual identification of these cells, just showing good agreement between the automated assignment of different cell identities in the islet compared to what has been previously observed there. And this is just an example of questions that we can start asking, where the different types of cells, there are alpha, beta, delta cells in the islet, where they are located and how they are connected. Again, this is just a kind of example of the type of analysis we've been doing. And what's gonna come next is really combining this with mechanistic models of about these cells and simulating this over time. Moving to our next major theme associated with antibiotic resistance. In the hub, this being led by Robert Birno, who has been working in that space for many years, and our postdoc Emily Wood. She's leading on this work, and I'm going to show a couple of examples of the work that they've done to date. Again, very similar to some of the other talks that we saw here, where they have started by leveraging on the wealth of information about the effect of antibiotics as measured by this minimal inhibitory concentration. So they have access to a database of 6.5 million clinical minimal inhibitory concentration for specific antibiotics. And what they're doing very often in practice, patients are given combinations of antibiotics, and in the absence of patients, meaning individual patients, there is no information about individual patients' minimal inhibitory concentration. This is the amount of antibiotic that will be effective against specific infection in a particular patient. So basically, the idea is here that by studying this wealth of information, some more general interactions between different antibiotics could be identified using various nonlinear measures such as mutual information scores. This is an example of two different antibiotics and their interactions in terms of susceptibility and resistance. And the idea is to take this forward, building a model that can help clinicians choose the combination of the drugs for individual patients. Again, this is only the beginning of this work. Further challenge with infectious disease and treatment is the fact, as you again all aware, that antibiotics and microbes develop resistance against antibiotics, and that happens quite quickly. So here the question is, can modeling help this process of selecting for resistance within patients rather than in the lab? At present, it's done in clinic, it's done using laboratory assays. So basically, by studying in more detail longitudinal samples from patients, the idea is to come up with some mechanisms that are associated with that resistance within patient samples. So this is a collaboration with University of Sydney and the hospital there. The postdoctoral fellow, she's very gifted. She also can perform experiments. So she's using nanopore technology to sequence patient samples, longitudinal patient samples taken over sequential over time and over different treatments, and then coming up with some mechanisms that then can feed and inform the modeling. That is the idea here. And one final example, this is a collaboration, international again, with NTU in Singapore and with a clinician and the student that we are co-supervising jointly together. Here we're looking at respiratory disease and combining data from different types of microbial communities. In particular, we have done so far work on integrating data from bacterial, fungal, and viral species. So our student is looking at developing this framework for integrating data and for using this in clinic. And I have one example here of how we have applied that to looking at the effect of antibiotic treatment. Here we have 12 patients that have received a specific class of antibiotic, beta-lactam, and they have been treated for worsening of their conditions. These are patients with bronchiectasis, so that worsening is called exacerbation. So this is basically an integrative network on interactome, as we call it, of different types of species. We have fungal, bacterial, and viruses here as well. So this is healthy. This is a stable patient condition. This is a chronic disease, so there is no such thing as healthy, but it's stable. So they, when their condition worsens, they've been treated with beta-lactam, and to simulate this kind of treatment, we basically change the amount of some of the pathogens that are known to be targets for these antibiotics, and then compared to a patient's network, and we find that 10 out of 13 interacting taxa could be predicted well. So the idea here is that by being able to identify which pathogens are affected by antibiotics and what interactions are changed, that can inform better treatments and better dosage of this type of also combinations of different antibiotics. So just like to finish with this slide, and this is really reminiscent of what Carola showed in the beginning. Basically the idea here is that holistic approach that integrates data from different type of, in this particular case, we're looking at omics data, and leveraging their relationships can really help in what clinicians call endophenotyping. that then ultimately translates into better therapies, better prognosis, and better diagnosis for patients. And that is one, although here it's specifically aimed at respiratory conditions, I think that's a common mathematical challenge behind this and methodological challenge, which is really common from all the presentations that I saw so far. Just here finishing with a couple of events, like all the other hubs, we have been engaging with the public. This is a public lecture that we held in March, and we have also hosted in June the Mathematics in Life Sciences meeting, where a lot of different aspects of applications to biology and medicines were discussed. And I know that next year there is again a joint initiative of a workshop that will be organized on uncertainty quantification between the UCL Exeter and Glasgow hub, I believe, in the Lawrence Center in the Netherlands. So with that, I'd like to finish, and I can take maybe one question. Thank you very much for your attention. Well, or more questions than one.
Thank you very much, Krasimira. Are there questions? Okay, I can start with one, Krasimira. So you mentioned missing data at the beginning, but I'm sure this happens in many of these projects that you have presented.
Yeah.
How are you dealing with this?
Well, we have been looking at imputation, actually. You're absolutely right. But that is a whole big topic by itself, how best to impute data. And in that specific project that that I mentioned, the model actually allows us to, we can use that model-based approach to kind of, because the model is continuous and predicts whatever resolution we choose. So we have been exploring how well imputing using the model performs compared to some other imputation techniques that are more, I mean, this is also statistical, but it's a bit more sophisticated. So I'm not sure if you're doing anything about imputation in Cambridge.
Actually, I have been looking into imputation quite a bit. It would be nice to discuss a bit.
Yeah, that would be great, actually, yeah.
But it's interesting. So you use the model itself to do the imputation. Okay, interesting.
Yes, and with the model, we can kind of, because it's again Bayesian, based on Bayesian principles, we can get these ensembles of trajectories, and then we can split them into, we can assume that certain things are like truant, others are predictions. And yeah, we are doing these kind of experiments at the moment with numerical experiments.
Excellent. Thanks a lot, Krasimira. I think probably in the interest of time, we're going to go to Hao next. Thank you so much, Krasimira. It was really interesting to hear from Exeter. Thank you.
I'll stop sharing here. Thanks.
All right, next we